What are enzymes and their basic function? Enzymes are specialized proteins that act as biological catalysts in living organisms. They speed up biochemical reactions by lowering the activation energy required for reactions to occur without being consumed in the process. Unlike chemical catalysts, enzymes are highly specific. Each enzyme catalyzes only one type of reaction or a group of closely related reactions. This specificity comes from their unique three-dimensional structure, particularly the active site where substrates bind. The lock and key or induced fit model explains how enzymes recognize and bind to specific substrates. Without enzymes, most biochemical reactions would occur too slowly to sustain life, making enzymes essential for virtually all cellular processes. Next, we'll explore how enzymes catalyze biochemical reactions. Enzymes catalyze biochemical reactions through a multi-step process that dramatically increases reaction rates. The first step in enzyme catalysis involves substrate binding to the enzyme's active site. The substrate moves to the enzyme and binds to the active site, forming an enzyme-substrate complex. This binding can strain chemical bonds in the substrate or position reactive groups optimally for reaction. Next, the actual chemical reaction occurs, converting the substrate to product. This is where the enzyme's catalytic power is realized. Finally, the products are released from the enzyme, which is now free to catalyze another reaction. The enzyme remains unchanged and can repeat this process thousands of times per second. Enzymes can accelerate reactions by factors of millions or even billions by lowering the activation energy required. This dramatic reduction in activation energy is what allows life's chemical reactions to proceed rapidly at normal body temperatures. Enzyme activity is precisely regulated through various mechanisms to ensure biochemical reactions occur at the right time and rate. These include allosteric regulation, where binding of effector molecules changes the enzyme shape. Competitive inhibition occurs when inhibitors compete with substrates for the active site. In non-competitive inhibition, inhibitors bind elsewhere on the enzyme. Finally, covalent modifications like phosphorylation can activate or deactivate enzymes. Environmental factors also significantly affect enzyme activity. Temperature affects the kinetic energy of molecules and the stability of enzyme structure. pH alters the ionic state of the enzyme and substrate. Substrate concentration determines how frequently substrate molecules encounter enzyme active sites. This precise regulation ensures that biochemical reactions occur at the right time and rate to maintain cellular homeostasis.